Hello, am I audible?
Good morning, Dr. Bhattacharya. Yeah, you are loud and clear. So this is
Yeah,
Dipanjan.
I'm just trying to present the uh, talk so that you can confirm me whether you can see it.
Sure, sir.
Okay.
Yeah. Yeah, it's fine. You just need to put it in presentation mode. That's it.
Yeah.
Sir, can you please make yourself exit from the presentation mode so that we can start the initiation process actually.
Okay, no issue. Yes.
Thank you, sir. Uh, Madam, should we start?
Yes. Very good morning to all the speakers of today and the participants. Yes, you can begin. Please do.
Thank you, ma'am. So, welcome to the second day of 2 days virtual workshop on confocal microscopy and digital image processing. We aim to have the sessions like, first of all, we will hear an application part from an eminent scientist. Thereafter, we will move on to the hands-on training, how the digital image processing occurs and so on. We'll be learning in the second half. So, with that, we'd like to welcome Dr. Shubhendru Bhattacharya. So he is a PhD in the year of 2003 from Jadavpur University. Thereafter, he moved to Basel, Switzerland for his postdoctoral studies. He is presently a principal to deliver for his kind deliberation on applications of confocal microscopy. So, Dr. Bhattacharya, the floor is all yours.
Yeah, thank you. I thank the organizer and thank you all for the attending this webinar. So it is always good to be there and interact with people personally and communicate the science that we do and the techniques that we are talking about, how this is useful for the science. But anyway, this is good enough to also interact in this manner. So I will probably put my video off such that there should not be issue with the streaming and other things. Sure. And again, when go back to the discussion part or any interaction part, then I'll put my
no issue, no issue.
Okay. So right now I start to share the presentation. So as it has been mentioned that we do really follow some basic questions in cell biological aspects of microRNA-mediated gene expression in mammalians particularly, and imaging is an integral part of our studies where we use different imaging techniques to address these biological events. So I will gradually tell you how these useful techniques like confocal microscopy or high-speed imaging that has become useful in identifying some of the finer details of these events. So today's talk is on importance of cellular organelles in controlling the microRNA-mediated gene expression in mammalian cells and imaging the cell biological events. So before we go into the details of the gene process and its regulation by microRNA, I like to introduce you that the microRNA are usually acts on the and axon. But before that, you can think about mRNA, the 5' UTR and the 3' UTR, the non-coding parts and the coding parts. These parts conserved across the evolution and in sequence, and they do interact with different protein factors. And these protein factor, by interacting with either UTRs or coding sequence, determine the fate of the mRNA. And as there are different elements span over the entire UTR and the coding sequence that can interact with different sets of proteins, you can imagine there is an mRNA code. That means the mRNA that interact with different proteins are defined by the sequence that they have in the UTR and the coding sequence. So these different protein along with the RNA interaction ensure the dynamics and mRNA of the mRNA, and the condition specific RNPs are formed. Sometimes some of the protein interact in a specific context, some of interact in a specific region of the cell. So these regulatory protein, scaffolding protein, accessory protein, as they can be classified into, affect the translation of the protein from the mRNA it codes. It also affect the stability of the mRNA, as some of the RNA binding proteins are destabilizing the mRNA, some of them stabilize the RNA, and they also affect the localization of the protein and the mRNA. So microRNA plays an important role in this context. They are 22 nucleotide long regulatory RNA, and they are transcribed inside the nucleus as precursor like primicroRNA, which is a stem loop style structure, long RNA, and acted on two protein GGCR8 dorsal-protein complex in mammalian cells to process them to a shorter form called pre-microRNA. And this pre-microRNA then exported into the cytoplasm and processed in the cytoplasm by dicer TRBB protein complex to the mature microRNA. Double-stranded RNA then cleaved by dicer, and one of the strand of this double-stranded form, which is here in red, get incorporated into the Argonaute proteins, and this Argonaute microRNA complex then form the microRNP and target the target genes where the microRNA binds with imperfect complementarities and induce translation repression, and also sometimes induce degradation of the target messages. So there are thousands of microRNA operates in humans. Almost 50% of our genes are controlled by one or multiple microRNAs. They regulate different development, differentiation, apoptosis, so many things. And the expression is also regulated in human diseases, dysregulated in human diseases, that there are upregulation or downregulation of specific microRNAs happens during the disease development process. And as it's important player in controlling development, they are also a developmental and specific pattern in brain, liver, and the muscle, for example. So this process can be reversible. In the process of translational depression, the microRNA can be stored in some kind of structure called P-bodies. And these P-bodies you can visualize by the microscopic technique. At that time, we only use the normal microscopic fluorescence microscopic technique. To find there are some several dots that are positive for the RNA, and later we confirm these dots are called P-bodies because these also called certain proteins, and they form this kind of granular structure within the cytoplasm. But if you put the cell under stress, for example, amino acid starvation induced stress, this mRNA get relocalized from this localized body structures where they were replaced by microRNA, and now they become part of the cytoplasms and part of the polysomes or ribosomes, and they get translated. So this way the process gets reversibly regulated. And one protein we identified at that time called HUR, which is an RNA binding protein that interacts specific sequence of the RNA present on the UTR of the mRNA, can regulate the microRNA activity in a negative manner. That means when HUR binds, it inhibits the action of Argonaute microRNA complex on the same mRNA in a cis-acting manner and induce the translational derepression of the mRNA by the microRNA. So this way the process gets reversed. So one can imagine, as HUR is one of the factors that binds along with the microRNA to regulate its action in a negative manner, there could be hundreds of such factors that can either positively or negatively regulate the microRNA binding or activity in a cis-acting manner on the same mRNA. So that means the recruitment of this regulatory factor, like RNA binding protein X or Y, that act as a positive or negative manner on the microRNPs to activate their action. So in that way, the interaction of the multiple RNA binding factor on the UTR can determine the fate of an mRNA, and their recruitment to these UTRs can be driven by different signals. It could be an external signal or cues, or internal signal like growth signal that can affect the binding of these factors on the UTR, and that way reversibly act on the microRNP function. So what are the factors that we are talking about that control the microRNA activities in mammalian cell? That is the primary question that we address in the lab. To that end, we have several important research needs. For today's talk, I will focus on the importance of subcellular organelles in controlling the microRNA biogenesis and activity. That means when you're talking about the subcellular organelles, we can consider different organelles like endoplasmic reticulum, endosomes, lysosomes, mitochondria. So they are the players and they're known to have specific function in the cells. Like, for example, mitochondria is known to generate ATP and generate energy. But apart from this role, the mitochondria could have a different kinds of role in gene regulation process also. That has not been explored before, and we are in that way somewhere pioneer to introduce the concept that gene regulation not only get regulated at the transcriptional level or at a transcription post-transcriptional events like spacing and export of mRNA. It could be regulated at translation level as well, and this regulation of the gene expression, which is also controlled by microRNA, can get influenced by the subcellular structures like ER and endoplasm. How we can come to that kind of conclusion, I am going to talk about. So the cell biology of microRNA replication is not well explored. For example, I told you that exportin 5 is a protein that presents on the nucleus membrane that ensure the export of microRNA into the cytoplasm, the precursor microRNA, pre-microRNA, that are further processed by this dicer TRBP to the mature form. And then the microRNA get formed with the agonot and mature microRNA, and that ultimately induces the gene expression. But whether within the cytoplasm, it's a vast area. It can have different microdomains, the rich in specific organelles like the perinuclear space, which is immediate to the nuclear site, are very much enriched with the endoplasmic structure and so on. So whether they have any preference to have these events like dicer processing of precursor microRNA or micromedia depletion that happens specifically in different compartments or not. So where the microRNA acts on their targets, and does translation, depletion, and degradation occur simultaneously in a specific context, or it is very much different sites within the cell where these events actually happen. So to answer the question, the first things that we have tried to identify in an experiment where primarily use a totally biochemical approach without any microscopic things by inducing an inducible target mRNA and following its fate with time. And we find by biochemical fractionation, we can isolate endoplasmic reticulum and cytoplasm and nucleus separated in this manner. So we can find that there is a microRNP and the target mRNA are enriched on an endoplasmic reticulum bound polysome. So endoplasmic reticulums on surface, there are ribosomes that are known to translate proteins that primarily get targeted to the endoplasmic reticulum membrane or lumen, and then ultimately exported out of the cells as secretory proteins or get to the membrane of another organelles. So this is the way transmembrane proteins and secretory proteins are sorted. The mRNA itself gets translated on the ribosome present on the ER. But that concept gets changed. It has found that the ER-bound polysomes actually can translate proteins that not necessarily need to get targeted to to a membrane or get exported out. That some of the known cytoplasmic protein can also biochemical approach how these repressions and the degradation of the target mRNA happens in the same compartment or in different compartment after they get repressed on the endoplasmic reticulum attached ribosome. So the question, as I mentioned, it's still there. Where does the repression and degradation of the target mRNA happens? To address the questions, we have to have another biochemical way to separate out different organelles, and we did that. We used the biochemical way to separate out the organelles on an optic gradient. So we use this 3 to 30% gradient. One can separate the endoplasmic reticulum from the MVV or endosomes compartment, and we can find that several protein factors necessary for the degradation of the mRNAs are enriched in the early endosome or MVV fraction rather than the ER. So it suggests that the degradation may possibly happen on the another compartment, like endoplasmic reticulum, not on endoplasmic reticulum, but also on the in early endosomes or MBB fraction. So this study further continued with a with the inducible reporters, where you can follow the repression and the target RNA localization simultaneously. And with that, we can find there is a spatiotemporal uncoupling of microRNA mediated translation mutations and target RNA degradation that happen in mammalian cells. For example, the target RNA first get repressed and then get degraded or get relocalized to the different compartments. That is evident from these data that we are presenting here. The impairment of this MVV formation, the endosome formations, actually increases the mRNA stability, which further suggests that the repressions doesn't get affected by the depletion of the one protein called HRS, which is endosome biogenesis effector proteins. And but the repressions doesn't get affected, but the target RNA level actually get increased. So from that angle, we can say that these processes are uncoupled to each other. So again, this is a biochemical way of proving a basic phenomena, where we find that the spatiotemporal uncoupling of microRNA-mediated translation repressions and target RNA degradation happens in mammalian cells in a spatiotemporal manner. So one can imagine the rough endoplasmic reticulum where the translation repression occurs with the help of the microRNPs and the mRNA and other interacting factors. They affect the translation initiation process. And then it's go to the early endosomes where the ribosome falls off, the mRNA get decapped and deadenylated. And subsequently in the late endosomes, the RNA get chewed, the microRNP actually get recycled again to the new mRNA on the late endosomal surface. And Leishmania donovan, which is a parasite, actually affect this kind of endosome maturation process by targeting this HRS protein, which is essential for the endosome maturation. If we affect this protein, we can find that the target RNA degradation affected, micron recycling get affected. So this early endosome to late endosome maturation step is very important as far as micron recycling is concerned. So the questions remains how the organelle dynamics, because what about the organelles we are talking about right now, like endosomes, endoplasmic reticulum, they are not very static entities. They move within the cells and interact among themselves. So how this organelle dynamics can affect the microRNA compartmentalization process and its activity. These questions cannot be answered in a simple biochemical manner because biochemistry deals with some kind of static environment where we have the organelles get separated to each other in a biochemical manner, and that's it. So you cannot really follow the dynamics of the organelles with a real biochemical assess. And again, how the compartmentalizations ensure the cooperativity in the translation depression process is also an unanswered question. So these are the two things that cannot be answered in a biochemical assay. So we have to adapt in a cell biological assay. For example, the hypothesis put forward, for example, is the interorganellar interaction decide the gene expression. So if you have these two endoplasmic reticulum here, and early compartment there, and which is interacting among themselves, and where you have the mRNA on the ER, whereas the microRNA may be present on the MBB, and they should interact to get a gene repression. If they don't interact, the gene repression will be there, the mRNA will get translated, protein will be synthesized. But if there is a interaction, the organelle interaction is essential for this exchange of this regulatory factor that present on the different cytoplasmic domain represented by early endosomes or ER. They must interact then, and they should exchange this factor to get a repression process. And in this case, when the microRNA get transferred, then it should get repressed. The gene should get repressed. So organelle interaction is essential for the effective gene degradation. That's the hypothesis we put forward. So to that end, when we do a dual color imaging, for example, this mitro ER imaging, where we are showing here mitochondria in green, which is expressing a GFP tagged version of a mitochondrial protein, and the endoplasmic reticulum is expressing the DS red tagged protein of a endoplasmic reticulum origin. And then you can do the dual color imaging in a Andros confocal microscope, spinning this confocal microscope, and then subsequently use the image to analyze the organelle structure. So when you do acquire some kind of 3D image reconstruction after the jet scanning of different planes, the image captured at different planes, over time also you can use in some kind of 4D imaging in this time as well as the G stack as well as a different wavelength also. So you can image all these things all together in this confocal microscope and really deconvoluted the image. You can see some kind of, if you don't vary the time, just fix a one particular time or a fixed cells you use in this case, you can see that they are entangled to each other, the mitochondria and ER. So you can really separate them together to each other. And if you use another color to denote the interaction surface, if you put this in, in yellow, so you can see that this is the interaction surface. That's they interact. Same thing you can do with in the mitochondria and early endosomes. You can also see they are very much entangled to each other. And if we do the same thing for the ER to endoplasmic early endosome, we can get a similar picture. So one can imagine the interaction is very much there, and they do interact on the surface of each other, such that it's not surprising they should exchange the factors that's present on the surface of either endoplasmic reticulum or endosomes. And we are hypothesizing that this kind of interaction is driven by the mitochondrial interaction with these specific organelles, because mitochondria is known to interact or tethered to this endoplasmic reticulum, and they form some kind of MEMS membrane, mitochondrial associated membrane, which is nothing but a part of an ER surface or membrane that are interact with the mitochondria. And people use this kind of structure to explain a lot of biological processes like calcium signaling and other things to explain on. So to that end, how we come to this kind of model, that was an interesting story to look at. So as I mentioned, the Leishmania donovan is a parasite that infect the human macrophage cells or mammalian macrophage cells and reside within the macrophage in specific compartments called parasitophagal vacuoles. As these parasites are very interesting parasites and they live within the macrophage, they also alter some of the machineries within the infected cells. For example, what we find in the context of microRNP that with infection there is an increase of organelles, the biochemical data. We have increase in the microRNA content also. There is some kind of stabilization of microRNA happens after the infection occurs. So this is very interesting thing. So there is elevated total organelle protein and microRNA level after the infection occurs, and we also find that in microRNA interactions, the clinical level of mRNA follow. Some of the cytokines, like pro-inflammatory cytokines like TNF alpha, IL-1 beta, IL-6 goes down, while IL-10, which is an anti-inflammatory cytokine, goes up. So this is very characteristic of Leishmania infection context. We find that the Leishmania donovan infection usually results in elevated host ago microRNA level. And we also find this increased AGO2 is endosome associated. Again, we fractionate the cells based on the density of different organelles on a biochemical way, to on a gradient, and we can see that there is an increased AGO associations with the early endosomal part that happens during the Leishmania infection. So how the decreased, whether there is any interaction between the endosomes and the ER that can explain this kind of phenomena we are getting in the Leishmania infected cell. So when we compare with the non-infected cell to the infected cell, I just here some ER interactions, and you can measure the Mendel coefficient about this co-localization coefficient in that way. So when you find that if you enlarge this particular part of the 3D reconstructed picture, you can see there will be early endosome mitochondrial interaction not much affected, but ER endosome interaction also get reduced. So we use this microscopy data to capture the infected cell to conclude that there is a change in the interactions of different proteins or different organelles, more particularly on the surface of the membrane. So that's pretty much specific with the infection process itself. So why there is a mitochondrial ER interaction loss? That was the question we also asked in the LD infected cell. So we try to follow that what happened with the mitochondrial dynamics after the Leishmanian interaction. So this is a non-infected cell. The time-lapse imaging has been done for the mitochondria, again expressing the GFP protein. And you can see in large part how this mitochondrial changes over time. So this is 0 second to, let's say, in 60 seconds, you can see the whole mitochondrial structure get altered with time, which is very much obvious because mitochondria is very dynamic organelles. But if you do this with the infected cells, you will find the change will be much less. So again, I am probably missing some panels here, but it's actually showing that there is no much dynamics that are showing. So there is a reduced amount of mitochondrial fusion that we observe in the infected cells, and we are done with the live cell imaging part with the mitochondria. So we also then follow the mitochondria polarization status. If mitochondria has a membrane potential, and that membrane potential is very much important for the mitochondrial function. So that suggests that the mitochondrial depolarization is required for the mitochondrial stabilization. So based on that, identified another protein MFN2, which is a mitochondrial tethering protein. So this is the ER in the mitochondria, which is tethered together by the two groups of protein, MFN1 and MFN2. They interact among themselves. The MFN2 is very much specifically present on the ER surface, and they have the interaction of the MFN1 and MFN2 on the mitochondrial surface. And if we have a loss of membrane potential or expression of UCP2, that actually affects this kind of MFN1 and MFN2 interactions, and ultimately the loss of mitochondrial ER tethering. So we say MFN2 knockout MEF, we had MFN2 is not expressed, we can see the similar increase of microRNA activity. So detecting of mitochondria ER by MFN2 ablation increases the level of MIRX for compound components and concurs with the increased microRNP activity. So what is the consequence of this increased accumulation of AGO2 on endosomes? So we find that actually this reduced microRNA present on the endosome, AGO2 present on the endosomes, is actually prevent the microRNP recycling in the mitochondrial defective cells. So the new microRNP cannot be formed. So reduction in the recycling is due to reduced AGO2 dicer interaction. That is another observation we had. And based on that, we have that model where published in the MBOC that Leishmania dominant restrict the mitochondrial dynamics to enhance the cycling and microRNP activity in the mammalian cells, the macrophage cells that the Leishmania, and that's actually useful for the Leishmania for the survival. The whole story was covered in the MBOC. You can see the images. And if I go to the images, you can see there are these different colors like things. So you want to actually use a image processing platform, Imaris, to highlight the interaction points where he has shown that how these different ER, mitochondria, and endosome, they do interact with each other. Some of the interaction points which is strong are shown in different colors. Like for example, this white part is representing the ER endosome or ER mitochondrial interaction affect the microRNA effect and microRNA activity. So that's the nutshell how the techniques that we have used, live cell imaging, time-lapse imaging, and multi-color imaging for the same cells with high resolutions can lead us to this question. Obviously, nowadays it is more interesting to use some of the super resolution microscope that are already available in our institute, and we are also trying to procure another one with structure illumination techniques to allow us to go into the more detail about these interactions that we are talking about, this endosome in mitochondria actions or mitochondria ER interactions, and about the finer details of the structures and the alteration that we are observing and their connection to the microRNA functions. So that's the thing we are following. So in that context, I'd like to highlight one paper we published very recently in the Life Science Alliance Journal, which is a sister journal of three academic press, Invo Press, Force Spring Harbor Laboratory Space, and I think Rockefeller Press. So they work on this, and this is a very interesting report we found that retrograde trafficking of Argonaut 2 act as a repeat step for the de novo microRNA formation on the endoplasmic reticulum attached polysome. So when you're talking about the microRNA formation, the Argonaut need to get loaded with the microRNA. And in that particular paper, we have shown that it actually happened on the endoplasmic reticulum, and which involves the Argonaut 2 trafficking from another compartments. For example, as based on the data we already published in this MBOC paper I talked about, we have other kind of experiment done in this particular paper of LSA to show that this is the endosomal surface where the microRNA get detached from the Argonaut microRNA complex, and this Argonaut then are further get loaded by a specific event called phosphorylation of Argonaut, and then it get dephosphorylated also, but this is without microRNA now. So these microRNA detached Argonaut present on the endosomal surface need to get recycled back to the endoplasmic reticulum, where the Argonaut dicer complex uh, and get the Argonaut get loaded with the microRNA for further action on another set of mRNA. So this way the recycling of the Argonaut and the microRNA and the microRNP happens within the cell. And in this paper, we have shown how the mitochondrial interactions with the endosomes or mitochondrial interaction with the ER, for example, the MFN2 here also, can affect this kind of trafficking process. So we show that this retrograde trafficking, that Argonaut coming from endosome to the endoplasmic reticulum back for a new set of repression to occur, they require the mitochondrial interaction with the endosomes and the and MFN2 affect the process. For example, if we do a contour map of the interactions that happen between the wild type and the MFN2 negative cells, we can see there is a loss of interaction between the two organelles, AGO2 and AGO2 and ER, that we can see here, that happens in the MF into negative cells. Similarly, if we go to the AGO2 endosome interaction, we can see in AFN2 cell there is an increased interaction that again the Binder coefficient representing here. So reduced localization of A22 to the ER is not only evident from the biochemical assay we have done, but that is get confirmed with the microscopic data we obtained with the cells without the MFN2. So this way, it strengthened the conclusion we made with the biochemical data we had, and that way it's very useful. As I already told you, that how this compartmental mechanism of post-transcriptional gene regulatory machinery are controlling each other, that is a interesting thing, and that we are following. So the questions how this interaction loss is there, that and how why they happen, and where this microRNP mediated repression occurs and how they affecting the whole process are the three questions that we are primarily following nowadays in this particular context. So you can see the beautiful image of the ER and mitochondria, how they intermingle with each other that I already shown, and the domains that they interact. So these are all were all possible because of the good microscope we had, and technique processing platform we use to get these images. So these are always evolving every time. So we always look forward to the new techniques and new tools that allow us high-speed imaging. So here we had the microscope that are capable of doing multi-channel high-speed imaging with more or less high resolution, but that resolution is not that great that we like to expect, and that will be taken care in a new platform that we're talking about, this uh, uh, structure limited microscopic techniques that will allow the super resolution imaging of the thing without compromising the speed of the image capture. So that way we are probably able to get a high speed multidimensional multicolor imaging with high resolution. So that's the ultimate aim. So before going to end, I like to highlight a few of the other work that also involves some microscopic techniques. This has been done in the context of neuronal cell differentiations. So Shomi Pratonovis in the lab actually identified some of the structures, as I told you, about the P bodies. These are formed, they have the AGO and by DCB protein all get localized. And when the differentiation occurs, these bodies become larger in size. And we have shown that P body components are necessary and sufficient for the differentiation of the PC12 cell and sympathetic neurons. P-body factor activate a pathway called P38 MAP kinase pathway, MSK1 pathway to phosphorylate AGO2, and this phosphorylation of AGO2 is essential for this kind of neuronal cell differentiation. So again, the biochemical data she obtained was supported by a lot of good microscopic data we gathered that useful to conclude about the biochemical thing we gathered. So as it has been mentioned that my lab is also very much interested in the exosome-mediated transfer of microRNA between cells. So exosomes are very small vesicles that are formed by the mammalian cells, and that way they get traffic from one compartment to the other or one cell to the other. So this is the initial observation made by another group. Transfer microRNA and it can be very non-identical. So you can imagine that NADs or the endosomes that we are talking about is actually also the sites where the microRNA get dislodged from the albumin protein, and this dislodged microRNA get packed into some kind of small vesicles, and within this vesicle, within the vesicle, which is called a multivesicular bodies, when fused to the membrane, they release these small vesicles within them as exosomes or extracellular vesicles that can carry the message across the cell boundary, and in the new cells they become active also. So one can imagine this kind of regulation of microRNA export is an effective way of control gene expression, where we're also doing some biochemical as well as microscopic technique to follow the event. For example, this exosomal export process is very much affected by the cellular status. So we've done the experiment to suggest that the polysomerase restrict the microRNA turnover by preventing exosomal export of microRNA in growth retarded mammalian cells. The proliferative cells have a tendency to export out more microRNA out of the cells, whereas the senescent cells have a less tendency, but the cause was unknown, and we identified that the polysomal retention of the microRNA is the cause that happens in the senescent cells for the lower export of microRNA and higher stability of microRNA inside the cell. So I just like to highlight another aspect, that what happens to the exported microRNA, how does the intercellular transport of microRNA contribute in controlling gene questions that we try to follow. In this context, I already told you about the one protein HUR, which is important for the microRNA deletions and dislodging of the microRNA from the target mRNAs. They reduce by polymerizing with the HUR, but the question remains, what happened to the GPS microRNA? So it has been shown, just missing the slide maybe, but in another paper we have shown that this actually helps export possibly identified as HUR actually helps the export of microRNA after dislodging this microRNA, and the mechanism has been explored. Homolika in the lab explored the mechanism and identified how the microRNA get exported out specific manner where the HUR plays a major role, which itself get some kind of post-nodal modification, ubiquitinations happen again on a compartment-specific manner like endosomes to get this microRNA out of the cells. Not only the mammalian cell secrete this exosome, leishmania also do, and these leishmanial exosomes again packed with some proteases called GPC protease. They target dicer to affect the microRNA processing step in the animal cells or the cells they infect. For example, in hepatocytes, they target dicer to reduce the microRNA 1 to 2 expression to reduce the cholesterol level. So in that way, we can tell about that this endoplasm intra and intercellular exchange of epigenetic signal in terms of microRNA can contribute and maintain the gene expression homeostasis in animal tissue. So sometimes the microRNA, after entering into the system, can go to the isosome, get degraded. They can packed into late endosome and get secreted, or they can be part of the cytoplasm, get released and get part of the cytoplasm. So all those things involve cellular dynamics of organelles and their interaction with different among the organelles, which can only be visualized with the microscopic techniques. So where the biochemical data that we always gathering with these organelles can be substantiated or strengthened by the good microscopic techniques that we follow. So I'll probably able to highlight how the microscopic technique helped us to get into the more detail of the mechanistic understanding of the whole process at different levels. So we acknowledge the fellowships from different funding agencies to answer these questions. And these are the people who did the work. I mentioned the work of Banishika and Moina Bose little bit. And then I mainly concentrated on the Vivalito Chakraborty's work. I didn't have chance to some of the recent work we have done, but these are the work also done by other guys. Sobik also did some of the interesting work, as I mentioned. Komolika did the HUR part of work. And these are the people who are also doing some interesting work, and I will be able to talk about that in another meeting, another platform. So funding board is also very helpful, and I will be happy to answer some of the questions you may have related to the microscopic techniques or maybe unrelated. It doesn't matter, and I now be available for any questions that you may have in the this context. So I'm stopping my presentations. Will It will allow me to interact with you and some of the things.
Thank you, sir, for your extremely astute deliberation. I would like to present the floor to ask any questions that they might have. So if you have any questions, please switch on your audio and ask, sir. Is there anybody? Anyone? Anyone on YouTube?
Some comments in chat? No, not. So you can put the question in the chat box also. I can answer that.
Okay. Evidently not. Okay, sir. Sir, okay. If no one else has a question, then I have a question. You mentioned that your talk was about the microRNA activity being regulated by mitochondrial dynamics and interaction with various organelles, like endoplasmic reticulum in macrophages, and this is how they regulate gene expression. Sir, my question is this: can this pathway be targeted to modulate different immune responses, or is it only specific to Leishmania domain?
No, I think you are very much right that this process can be affected in immune responses as well. So, of course, when you're talking about the macrophage and the Leishmania infection, this is one way of immune response, where we have the anti-inflammatory response occurs predominantly. But it could be the pro-inflammatory response like LPS stimulation and other things that are also happening. And more interestingly, the activity of these mitochondria and other things also get affected by some of the pathogenic factors or non-pathogenic factors like phagocytic activity and the movement of the endosomes that are all getting affected. So, for example, when you take the cells and find their activation with the LPS, you find there is a loss of microRNA activity happens during the early phase of activation. For example, at first 2-3 hours, you have a total loss of microRNA activity happens in the treated cells after the LPS stimulation. And that we identified as a because of ago2 phosphorylation. So later we are now doing what? That the localization of the microRNA and the targeting to the specific compartment is actually getting affected during this LPS activation, and that may cause the gene expression pattern change, where mitochondria also play an important role, as it also one of the recent work we are working that how mitochondria affect the mTOR activity and by the way activate the microRNA pathway. So this way, these are very much interlinked, and not only in the context of Leishman infected cells, but also in the other context as well.
Okay, so I have one in the continuation of your answer. So if the macrophage is polarized to anti-inflammatory state with like IL-4 or IL-13, so then if you try to challenge them with Leishmania, so what fate might be you are expecting, or if you have any result?
Yeah, so that's a very interesting point to make. But sometimes we do find for certain stages of Leishmania that become more infective because of the anti-inflammatory nature of the cell. But sometimes it also hampers some of the stain to enter that we are getting. Some of the drug sensitive and resistant stains, we are getting a difference in the pattern of response. That's possibly because of some of the receptors that they present on the membrane are getting different because of the cytokine expression, essential also for the internalization. So that depends on the leishmanial stain you are talking about, but most logically and as expected, it happens mostly in the favorable manner, that if you have anti-inflammatory pathway on, that it favors the leishman infection. That we already observed. And one of the work recently published, I didn't have a chance to highlight on that, is a work we published named Molecular Medicine, where we actually showed 1-phosphatase PP2A, which is used by leishmania in the macrophage, which try to put the infection on by upregulating the anti-inflammatory genes, whereas the protein H1 that I already talked about, when expressed, they export out some of the microRNA out of the cells, which is a very much export regulatory protein, micron export regulatory proteins. So that caused the pro-inflammatory response to dominate. So if you have a cell with high HUR protein expressed, that driving the export of microRNA from the cells, and that way it ensures the pro-inflammatory response dominating. So this way the process gets affected by different pathways.
Oh, thank you. So one guy has posted something, I think. Nirajan Ghoshal, he is asking, I would like to know that which kind of software is used to measure the core localization or internalization between organelles. Is it associated with the microscope or available otherwise?
No, usually we always get some kind of software with the microscope with some of the module attached. But in this case, we use a very advanced one, which is called Imaris platform, and they have different kind of module that can be added on the main software, like for example, co-localization module and organelle tracking module, something like that. So these modules are used separately, and this is a little bit costly software to get. But we use that when you do this kind of image processing. And this Imaris platform is a very good platform to do this kind of analysis. The images obtained with almost all types of confocal platform, like it could be Zeiss, it could be Leica, it could be Endor, it could be Olympus, doesn't matter. But this image will be processed in a third party software, in this case Imaris, and you can get this image.
Thank you, sir. If anyone has any more questions, YouTube? No, guess not. Okay, sir. Thank you very much, sir, for your very interesting deliberation. We will now continue to the next part of our program, where Dr. Manoj Mathew will be delivering his lecture. Dr. Mathew is a PhD on biophotonics. He did his postdoc studies from the University of Gothenburg, Center for Molecular Platforms, National NCPS. He was a part of Sahartaya College of Engineering and Technology as adjunct professor. He was a facility in charge of the light microscopy core facility in NCPS, and thereafter he has migrated to Karzais, where he is the business developmental manager of digital services and software throughout Asia Pacific. So we will be hearing now the hands-on training on digital imaging processing from Dr. Manoj Mathew. Sir, the platform is yours. Thank you.
Thank you so much, Dr. Bhattacharya, and welcome everyone to this part of the session. So what I want to do here is to first give you a short presentation and a little bit set the stage for image processing, and then take you through some practical aspects, give you a short demo of some aspects of image analysis. So is my screen, can you see my screen?
Yeah, you just need to put it in presentation mode.
Sure.
Yeah, it's fine now.
All right. So my presentation is titled From Images to Discovery, machine learning to cloud computing. So I want to introduce few new concepts in image analysis, which is machine learning and cloud computing, while also touching upon a little bit on the standard aspects of image processing. So this is how this talk will be titled. I will start with basics of images and image analysis. Then I will talk about Intellisense, which is a machine learning based image segmentation tool. And then I will talk about Appear, which is a cloud-based image analysis platform for microscopy. Now, to understand imaging or image processing or image analysis, we should first understand what an image is, right? So if I ask this question, what is an image? So the simple answer is image is intensity distribution over space. So anywhere you see in space where you see that intensity is varying, there is an image. So this could be a painting, this could be what I see with my eyes, this could be a picture captured by a microscope, or this could be a picture captured by a mobile phone. So anywhere where you can represent intensity as a function of space, there is an image. Now, if intensity is a function of space, the best in an image, the best way to represent that is as an array and as an array of elements. And these elements, some of you may already know, is called a picture element, or what we generally call as a pixel. So I have an image here, and if I zoom into this little region here, you can start seeing the individual elements. So these elements, as we were saying, is called pixels. And a pixel has a fixed intensity value. That's how you would associate or represent a pixel with. And this intensity is represented by a number. So here you see the pixels, and here you see the corresponding numbers for these pixels. As you can see, the brighter the pixel is, larger the number is. And darker the pixel is, smaller the number is. So this is a very intuitive and simple way to represent an image. And this kind of a representation is called a bitmap. A bitmap because you convert these numbers into bits to store in a computer, and then you have a map, which is like an array. So you have an array of numbers, which is what image is for a computer. Now, this would get a little bit more complicated if you want a color image, because color has to be represented as a sum of three different colors, let's say red, green, and blue. So pretty much any color can be represented as a linear combination of red, green, and blue. And if you have a color image, then instead of one array, you will have three arrays, which we'll call R, G, and B. A very important aspect of an image is called the metadata of the image. So what is metadata? Metadata is all the information in that image that is not image itself. So this is like additional information that is embedded into the image. So what kind of information are we talking about? We're talking about information like what filters were used for fluorescence imaging, what was the laser power if you are using laser scanning microscopy, what was the exposure time of the camera. And if you look at it, there are hundreds of parameters associated with the instrument itself. Now, in this, the most important parameter is called scaling. Scaling is a way of connecting or relating the pixels in the image to the real world. So in this example, you can see one pixel is 0.227 micrometers in the X dimension, and a similar value, 0.227 micrometers in the Y dimension. And if I have this information, then I can start doing real world measurements on my image. So based on how this metadata is embedded into the image, you have different image formats. So the common image formats that you come across, BMP, JPEG, PNG, they don't embed a lot of metadata. And that is why for scientific applications, you may not use the standard image formats like BMP, JPEG, or PNG. You need to use a more scientific format for storing data, for saving images. And a very common format is called TIFF. TIFF is a format that can implement metadata, and that is commonly used across multiple microscopy platforms. Of course, each microscopy company has its own standardized platform. For example, the Zeiss microscopy format or image format is called .czdi, and it can store around 476 different metadata entries. That doesn't mean that every image will have 476 entries. It means it could store up to 476 metadata entries into the image metadata part. All right. Now, let's look a little bit at the journey of a microscopist. So if you are a biologist especially, and if you are doing microscopy, you will know that that journey is quite difficult. And we already saw in the previous talk a little bit about how you go from images to data and how generating that data can be challenging. And that is what finally gives you the information you want. So you usually start with sample preparation, then you acquire images on a microscope, then you have the images with you, you have to segment the image, then you would do visualization, you would do plotting or sorting of this data, you would want to do sharing and feedback with your colleagues or collaborators, and finally there is interpretation. And this is a very long process. So between imaging and finally interpreting that data, there are a large number of steps, and these steps are usually falling under this umbrella of what we call as image analysis. So what is image analysis? According to Wikipedia, image analysis is the extraction of meaningful information from images. So here I have a simple workflow that shows you how we do this. We of course start with an input image. So this input image could come from a microscope or some other imaging device that you have. And this input image has now to be pre-processed. The pre-processed image is then segmented, and then finally the segmented image is analyzed. Let's take a very simple example. Here I have an image of some coins. This image is now pre-processed in the first step. So if you could guess what the pre-processing steps are, the steps are conversion from color to grayscale. And if you can observe it, I'm not sure if it's clear, we've also done a little bit of low pass filtering. We have made the image a little bit smooth. Why did we do this? To make this process called segmentation a little bit easier. So the pre-processed image is segmented, and here the image of coins are segmented into two groups, the small coins and the big coins. The small coins are indicated with the red color, and the big coins are indicated with the green color. Once you have segmented, now you can start doing measurements on these segmented objects. And there are a large number of measurements you could do. You could measure the area, you could measure the diameter. If it's a three-dimensional object, you could measure the volume. You can also measure things like ferret ratio, the major and minor axis of an ellipse that can be fit onto the object, and so on. So there are a lot of measurements you can do once the image is segmented. So let's take a little bit more closer look at the individual steps. So once you have the image with you, the first step, as I was saying, is called image processing. I don't know if you can recognize the gentleman in this image. He's Ernst Abbe, who is one of the pioneers in modern microscopy. The first image that you see, that's the original image. All the other images are being processed in one way or the other. So this image, for example, has been converted from grayscale into a binary image. This image has been geometrically transformed. This image has been undersampled. This image has been low-pass filtered. This image has been subject to what is called as edge detection. And this image is, in fact, a Fourier transform of this image. We have converted the image from let's say, the spatial domain into the frequency domain. So quite a lot of image processing steps you could do before you do the main and the most critical step in image processing, which is called segmentation. So what is segmentation? So as I was saying, this is the most crucial step in image analysis. Segmentation is about detecting objects against the background. Here you see we have cells. In this image, there are cars. In this image, there are some circuit board or a semiconductor image with certain pads. So these are the objects of interest. And in these images, we see, the objects have been extracted or objects have been detected by the computer against the background. So if you want a more scientific or technical description of what segmentation is partitioning of a digital image into multiple segments. So you're basically finding groups of pixels that belong together. The end process of segmentation is what we call as a binary mask. So here you see, if I have something like this violet region is where you have a car, and that is a binary mask. So wherever you have the car, let's say the bit value will be 1, and the remainder of the regions, the bit value will be 0. And if you're detecting multiple objects, there will be multiple masks. So each object will be associated with a particular mask that will tell you whether the object exists in the image or not. Now, how do we do segmentation? The traditional way of doing segmentation is what is called as threshold-based segmentation. And this is something you can do very easily in an image with very high contrast. For example, what you get from a fluorescence microscope, where the background is dark and the objects are really bright. So I can very easily tell the computer that if the pixel intensity is greater than a certain value, then it is part of the object. If the pixel intensity is below a certain value, then it is part of the background. So this is a threshold that we are talking about. Above a threshold, it belongs to the object, and below the threshold, it belongs to the background. It's a more, let's say, straightforward way of segmentation. Now, this may not work for all kinds of images. For example, this image here, if you observe carefully, the difference in intensity between the object and the background is not so much. But the variation of intensity within the object is much higher. You see, the background is more or less smooth. The object has more variations of intensity. So instead of looking at intensity itself, we can look at variation in intensity. So that is called variance-based method for segmentation. Now, in these two methods, we can see we use just one parameter to segment the object. In this case, it was intensity. In this case, it was variation in intensity. But if you have a very complex image like this, segmentation based on just one feature or one parameter may not work. And that is where you might want to use machine learning based segmentation. And this is what I want to talk about briefly in the next part of the talk. So at Zeiss, we have developed a tool called Intellisense, which is a tool to perform machine learning based segmentation in a rather easy to use manner. So what is machine learning? So we know humans, we operate by learning from experience, examples, and interactions. That's how we learn. But machines or computers usually work on hard-coded rules and instructions, which are called as algorithms. So traditionally, computers have worked very differently from the way humans have worked or humans have learned. But the question we want to ask is, can machines or computers learn like humans do? Can machines and computers learn from experience, examples, and interactions? Why do we want that from machines and computers? Because we want machines and computers to solve complex problems that are complex and diverse. And we want machines that learn from experience and derive the rules to solve the problems by themselves, which means we are subjecting them to certain inputs, certain experiences, and they kind of develop this algorithm by themselves. So this is maybe a very crude way of explaining what machine learning is. So by learning from experience, what do I mean, right? So all of you in this audience can very easily identify that this is an apple and this is an orange, right? But for a computer, this is not very easy. It's a complex task for the computer. So as a human being, how do I know that this is an apple and this is an orange? That's because when I was a child, somebody taught me, maybe my mother taught me, my mother showed me, okay, these are apples, these are oranges. And then I started to learn that. And whenever I see an apple or an orange, what is my brain doing? In fact, this is happening subconsciously, right? I'm looking at the color, size, shape, texture, shine. So I'm not just making this decision based on one parameter or one feature. I'm making this decision subconsciously using a large array of features, right? And that is how we humans work. That is how we humans differentiate between objects. So humans learn to associate features with objects. So if you are using machine learning for image segmentation, this is what we want to do. So machine learning changes the way a system or a computer solves a problem. From a mathematical logical approach, we want a more natural and observation-based approach, which uses data and which uses experiences. So a problem with traditional machine learning. So machine learning has been around for a very long time. People have been using it for segmentation. And the difficulty is, if you are using machine learning, you need some kind of a programming skill. So you need programming skill to train and create what is called as a trained model. And then you need programming skills to use that trained model to actually segment the image. So traditionally, machine learning has been the strength of people with good programming skills. But this is what we try to change at Zeiss. We wanted to create a machine learning based tool that can be used by anyone without having to really know what really goes on behind with the complex algorithms. And so the core algorithms that work in Intellisense is something called scikit-learn, which is a package with Python, and another one which is called TensorFlow, which is from Google, and which is now one of the most advanced machine learning algorithms used by almost everyone who is developing applications on machine learning. But with Intellisense, all these complications are hidden in the background. What the user gets is an extremely easy to use interface. So you get something like a paintbrush. So the paintbrush, in fact, means it is something like Microsoft Paint, right? So you use your paintbrush to paint here, say, this is the background, you paint here to say this is the object, and so on. So it's very convenient and very easy for a user to interact with this interface and train the machine learning algorithm. Once it is trained, it creates a trained model, and then you can proceed to segment your images. So usually what you do is you train few images, and once these few images are trained, you can start segmenting the untrained data. And this is where machine learning becomes very powerful. So Intellisense is extremely easy to use, and it makes it easy for non-experts to use machine learning. Here I have an example. And if you look at this image, this is a quite challenging image because there is a lot of background, right? The background itself is quite bright and it's non-uniformly bright. So if I were to use intensity-based segmentation, the traditional threshold-based segmentation, this would not have actually worked. But with machine learning using Intellisense, you see we were able to segment this very clearly. Another example here, these are yeast cells. So as you can see, there are two groups of yeast cells, one that are floating, the other one that is stuck. And again, this is something difficult to do using traditional image processing or image analysis methods. But with machine learning, we were able to do this rather quickly or rather easily. So here I have an example or a video that shows how Intellisense works. So the user is painting and showing where the background is, where the cells are, and the user clicks train and segment. So this model is saved now. Once segmentation is done, we can use it for image analysis, extract this kind of detailed information from these images. And Intellisense is quite diverse in the sense that it can work on all kinds of data sets. It could even be an image that comes from your mobile phone, but it has been mostly fine-tuned for scientific images that comes from an X-ray microscope, a light microscope, or an electron microscope. Now let me quickly move into the final part of my presentation, which is using the cloud to do image analysis. So this is something really new. There are very few tools available right now where you can use the cloud for image analysis. Now, why do we need a cloud-based platform, right? So this idea came about when Zeiss was talking to many of its customers, and we asked the customers, what are the challenges you face as a microscopist? And there were many different answers and very, very interesting ones. So some people said, one challenge we face is we use very different pieces of software. We use one software to acquire the data, another software to maybe segment it, a third software to do complex analysis, and even a fourth one to generate graphs and charts and create visualization tools and so on. The next challenge, which is increasingly being faced by biologists, is the off-the-shelf software that you have, like we have from Zeiss or Amaris or Arrivis or once the Intellisense we have from Olympus. They all do a standard set of image analysis, but very often you end up with problems that cannot be solved with standard image analysis. And many labs now have to hire experts in programming so that specific algorithms can be designed. But then not every lab has the luxury of having programmers. Even if you have programmers, the next problem is different groups use different programming languages. So for example, I like Python as a programming language. You might like MATLAB. And if I have written a script to segment images, and if you have written a script to analyze segmented data, we cannot really collaborate with each other unless one person converts all the code into the other platform. The next problem is there is a lot of manual work involved. And if you have been a student working with images, you use Image or some other software, you click and click and click before you can get the final output. Now, this manual work really means that different students, if they are asked to do the same image processing task, they will come out with different results on the same data set. Which means reproducing published results is a nightmare because you cannot really tell someone what exactly was the manual work you did. What were exactly the parameters you set? How exactly did you move those sliders to get those optimal values? So telling how you exactly processed your image becomes a challenge. And finally, there is always a problem. There is one PhD student or a postdoc who knows how to analyze or process images, and once he or she leaves the lab, then the code that they have written or the workflows they have developed will remain in some computer, and the new person has to start all the way from scratch. So that is a set of problems. The other problem is computation infrastructure. As images become complex, as image, let's say, analysis becomes more and more advanced, we need more advanced computation infrastructure. This means you need better CPUs, we need GPU clusters, we need storage servers, and so on. This is expensive, and not everybody can have access to this. These are some of the challenges that we want to solve with this cloud-based platform called Appear. Cloud, as you know, it means that you don't do anything on your own workstation or your computer. You connect to the cloud using a web browser, and everything is happening on the cloud. So this is a cloud-based platform for image analysis, basically designed for microscopy. And this tool is called Appear. And with Appear, you can create and customize image processing workflows. You can automate workflows to increase productivity. You can share your work with individuals or the entire community. And this creates a very good platform for you to create end-to-end workflows from images to final output, and then share it with the collaborator. So how does Appear work? You log on to www.appear.com. You pick some blocks which are called modules. A module is a piece of software that performs a single task, so which could be like conversion of a file from one format to other, or segmentation, or doing a specific analysis on segmented data. So each block has a specific task. And you assemble these blocks together into a workflow, and then only thing you have to do is run the workflow. And this is done on a web-based interface or a web browser-based interface. This is how it works. So here the user is assembling the blocks. Then the user is connecting these blocks together. The output of one block is connected as the input to the second. And then we save the workflow, and then we can run the workflow. So if you look at it, it's a little bit like Legos for image analysis. You know Legos, right? So you have these little building blocks, you put them together and you get something functional out of those building blocks. So a very important advantage of Appear, and this is probably the only tool currently available in the market where you can create a workflow with different programming languages, right? So here I have a workflow where the first module is written in Python, the second module is written in Java, the final module is written in an ImageJ-based macro, and so on. So what is happening is once a code is written, this code is kind of encapsulated into a container, and this container is provided by a technology called Docker. So we have this container that is fully encapsulated. So when you run the workflow runs from left to right. When the engine reaches the Python or the execution reaches the Python module, it uses a Python engine. And when the execution reaches a Java module, it uses a Java engine. But the Python module has given certain outputs to the Java module, and the Java module is giving some outputs to the image module, and so on. So the outputs that each module provides could be an image, could be an Excel file, a CSV file, and so on, which the other module can understand. So this way, if I have written a program or a code in Python and you have written a code in Java, we can in fact collaborate with each other. And this also helps users to create end-to-end image analysis workflows from images to the final results. And whatever you have created can be shared with somebody else. So you can choose to share it with your collaborators. You can keep it private, of course, or you can choose to make it public, which means you publish the module, get the whole community on Appear use these modules. So Appear is a little bit like a community initiative. It's a little bit like Wikipedia, where users contribute content and other users can then consume the content. It's really a community initiative. All the data that you generate, even intermediate results of different modules, are stored on the cloud. So you can go back, check how the image workflow has worked. If there are any problems, you can fix it. And if you don't want all this data, you can easily remove it out of the cloud. And it also makes it very easy to share the data. So we use an interface called Arravis. This is a web interface to visualize your images. You can even do 3D rendering on the cloud. So in fact, if you have a 3D data set, you could do 3D rendering on your mobile. Otherwise, to do 3D rendering, you would need something like a GPU support. This is an example workflow, and this workflow was created, I believe, for a university in the US. And what they were doing is something called as a scratch assay. So scratch assay, many of you would be familiar. You have these cells, you throw a scratch with a tiny pointed knife where the cells are removed. And then because the cells are growing, they slowly fill this gap up. So depending on how quickly and the gap is filled up, you can measure, how fast the cells are growing. And this kind of studies are done very commonly for wound healing studies and so on. So this was actually being performed in a manual way by the research group, but we were able to automate this using Appear. So we have 4 modules here, one module to acquire the image, the second module to do some pre-processing, which is in this case, adaptive histogram equalization. The third module, the most important step in image analysis, segmentation, and the final module to generate the report. So in fact, pretty much with one click, they go from images to data using Appear. Here I have an example, a video that shows you how Appear works. The user is assembling different modules. This is a cell counter and analyzer assay. This is a pre-processing step. This is a segmentation step. This is a post-segmentation processing step. And this is a report generation step. So once the modules are assembled, they're connected together, the file is loaded. You see, all this is happening through a web interface. So you can see here, this is all on a web interface. There are some parameters that you'll have to set for each module. And then finally, you run the workflow runs from left to right. And this is something you can even run on your mobile phone. You don't need any software. You don't really need any advanced computational hardware. Only thing you need is connection to the internet and a web browser, something like Chrome. Okay, so that was a quick overview I wanted to give you before I start with some demonstrations of how we could do, how we can do image analysis. So if there are any questions, maybe we can take those questions first before I go into the demo. Any questions?
I think there is a participant, Saurabh Ghosh and Niranjan Ghoshal. They have wrote for you something in the chat box. So I'm just reading it out. If I am looking for any protein foci within cells by immunofluorescence, is it the software able to count the number of foci? Can it distinguish between two overlapped foci or closely pointed foci? Hello?
Yeah, so the question is for a specific image analysis problem, right? So you have multiple components, and the question is whether you can differentiate between one component and the other. So image analysis, as I was saying, it is a workflow that you have to create, right? So whether a particular software can do this or not really depends on how you can configure the workflow there. So in your particular example, you might want to segment the two objects separately. And again, without actually seeing the image, that will be a bit difficult to tell. But in a software like that have shown or I've discussed about, it's possible to segment two different categories of objects out. In fact, one of the demo I'm going to show you is exactly that. How do you identify two different groups of objects and then do analysis separately on these groups? Yes, it is in principle possible, but again, every image is different. And unless you actually take a look at the images, you cannot tell for certain. Is there another question?
Yeah, I think so. So another is the scratch assay data you have shown. I assume it is the time course image.
Yes, it's a time lapse.
Yeah, there is a second line. Can I use this with two different endpoint images?
Two different endpoint images. I'm not exactly sure what you are referring to.
So I think he's hearing you. So you can come ahead and ask him the question directly. I think he is not here. Anyway, so there is another. Can images captured by other microscope be used in Zen installers or in Appear?
Yes. So Zen has options to we have a tie-up with OME. OME is an open microscopy environment. It's a university initiative from University of Dundee, and with this initiative, microscopy formats from different companies can be converted into a standard format, and this is called OME TIFF. So we can, in fact, read formats from other microscopy companies. Again, subject to the agreements those specific companies have with this open microscopy environment society. But pretty much most of the formats that we have come across, we are able to read it on Zen. And I'm sure this is also true for other companies. Other companies would be able to read the CZI format from Zeiss, again using what is called as bioformats through this open microscopy environment society.
Hello. This is Nirajan. Hello.
Yeah, Nirajan.
Yeah. I have measured. I have referred in these two different endpoint images like that. If I am taking an image at the first at the onset of my experiment, and after like 24 hours or 12 hours, I have another image where the cells are overlapping or not. So I am referring that these two images, can I use these two different time point image can be used in this scratch assay module?
Oh, okay, I get the question. So, so the question is, the one I showed was a time-lapse image, but you have two separate images with two
Yes, sir.
separate endpoints,
Yeah,
right? Yes.
yeah, yeah,
We
yeah,
can do
yeah.
that. But that exact workflow will not work. We have to create a slightly different workflow for that. But it is possible.
Okay. Thanks a lot.
All right. So if there are no more questions, then maybe I move on to the demo part. Is my screen still visible?
Yes, it's fine. You can go ahead.
So the screen that I'm showing you now is the software called Zen, and I'm using Zen as just a demo tool right now. So what I'm showing on this platform is also possible on several other platforms from other microscopy companies as well. So the problem that I want to solve today is, as you can see, I have okay, I can hide this. All right. Okay, I have this time-lapse image here. As you can see, this we have image nuclei are DAPI stained, and this is a time-lapse image, right? So I'm going to solve a very simple problem, and which is one of the most standard problems in image analysis. Well, that is to detect these objects and classify these objects into different categories, and then to do some measurements over time, right? So detect objects, classify objects, do some measurements over time. A very standard problem in image analysis. So what I'm going to do is I'm going to create a workflow. So the first step is creating a workflow. Now, why do I want to create a workflow first? If you have been using ImageJ, for example, you know that every time you acquire an image like this, a time lapse, you would repeat your steps, right? Okay, you will do the first step, pre-processing, segmentation, analysis. So next day you acquire another time lapse, you repeat the steps. And if you have 100 images, you repeat the steps again and again. You keep clicking and clicking and clicking, right? So what we want to do is to save that process. Do this only once, and in this process, create a workflow. Once the workflow is created, you can do this analysis with one click every time you have a similar kind of an image, right? So I'm going to create a workflow, and the first thing I'm going to do is I'm going to give it a name. So I'm going to call it cell analysis. Use any name. Okay, that name is already taken. I call it cell analysis 2. All right. Then I'm going to say set up my image analysis. So this is a guided process. Guided process in the sense that this software actually takes you step by step in setting up the workflow. The first thing it is asking me is how many classes do I have? So by classes in image processing means how many sets of objects do I want to detect? So typically there is only one color, and mostly people will say, okay, there is only one kind of object. But even within this one channel, I can detect different classes of objects. So let me try to detect, let's say, round objects, and add another class which is called elongated. Now, if you look at the image, some cells are slightly rounder, some cells are slightly elongated. There is not big differences between round and elongated, but we can set some criteria to determine which are the rounder ones and which are the elongated ones. So I have defined two classes or two groups here. Now, for every group, there is a super group, right? And so our group was called a class. So I gave a name for the class, and the super group is called classes. What is the super group? I will tell you later once we are done with this workflow. So let me call this round group, and let me call this elongated group. We already got some hint here. The group class, go back. The group class is doing a group measurement. It's doing a global measurement. And the class itself is doing a local measurement. What do I mean by that? So if I measure area in the group class, it is telling me the area of all the cells that belong to a certain class, all the cells in that class that is in the frame. If I go to the class itself, it is telling me or giving me area of each individual detected objects. So class is for individual objects and group is for group measurement in a given frame. We'll see that more clearly as we progress. Okay, so we have set up two groups, round and elongated, that is defined. And I've also given them two colors. I've given a red color for the round group and a green color for the elongated group, just for us to distinguish them clearly. In the next step is asking me whether I want to analyze the whole image or only a small region of the image, right? So this, yeah, I'm sorry, I live near a air base in Singapore. So they have this sortie, so sorry about the noise. So in this step, all right. So in this step, it's asking me whether I want to analyze the whole image or a region of interest of the image. So I can draw a region of interest like this. Now, why would you want to create a region of interest? So many times you will realize when you acquire the image, the actual region where there is activity, actual region where you really want to do measurements is confined to a small region of the image. If that's the case, you can always select a region of interest, which means the processing time for image analysis would be much smaller. That is one reason. Second reason is when you acquire an image like this, there might be some noise in the surrounding regions, and noise will interfere with image analysis many times. So you might want to avoid noise and keep your image or analysis area limited to where you really want it. In our example, we are not going to do that. In fact, if you want it, we can do it at a later stage. So that is why I've clicked this interactive button, which means even after completing the workflow, I can still decide whether I want to select a region of interest. I'm not doing it at this point. The next step is in fact the most significant step. Here is where we will do all our pre-processing, segmentation, and post-segmentation processing. So let me go to the first class, which is the round class, and do some pre-processing. I'm going to do a little bit of smoothing. So the cells are smoothened a bit. It's a bit difficult to see here. And once you have done the pre-processing, you do segmentation. And here there is a very easy automatic tool to do segmentation. And I simply click automatic. And you see all these objects were segmented. Right? You see the red color here. And we have different algorithms to do segmentation. Otsu. Otsu is actually the name of a scientist who developed that algorithm. And Otsu is one of the most common intensity-based segmentation algorithms. And there are other few algorithms as well. For every algorithm, there are two flavors. You have the light regions option or a dark regions option. What does that mean? It's asking me whether my objects are light against a dark background, like a fluorescent image I have. So it's a light region. You might also have a situation where your objects are dark against the light background, maybe something like phase contrast. So I've selected this Otsu segmentation using automatic segmentation tool. Light regions, I clicked automatic, and then all these objects were segmented. Now, very often you see the objects are so close together that they start touching each other, right? Because they're so close. Now, when this happens, the segmentation algorithm thinks this is one object. Now, there is a way to separate them. And one of the very commonly used algorithms is called watershed. And you see when I use watershed, these connected objects were broken into two. So this is giving a little bit more fidelity into my image analysis algorithm. Now I have to do the same thing with the other class. So I'm going to do low pass filtering, automatic segmentation. I'm going to use watershed, which is doing my post-processing segmentation. So again, let me recap. We used pre-processing step, which was low pass filtering, segmentation step, which was O2 based intensity thresholding algorithm, and a post segmentation processing, which was watershed algorithm to break these objects into two. Now, if you look at these two classes at this point, you see there is no difference between the round class and the elongated class, right? So my aim was to separate them out. That's because I've still not defined a criteria for round and elongated. That's exactly what I want to do in the next step. So here I'm going to give some morphological parameters. I'm going to tell the software that based on these morphological parameters, I want to select or filter certain objects. So the round class, I'm going to filter it based on area. And I'm going to use, you can see there are maybe 100 parameters. I'm going to use a parameter called circularity. So what does circularity mean, right? So if you have an object that is a perfect circle, circularity is 1. If you have an object that is a perfect straight line, circularity is 0. And our objects are always somewhere between 1 and 0. The moment I selected the criteria, the software has kind of disabled all segmentation, because right now I'm saying I want objects between area 0, 0, 0 and 0, and circularity between 0 and 0. So it's not detecting any objects that fulfill this criteria. Now I can, of course, fill in the criteria here. I can say I want to detect objects between area that is 100 micrometers square to 30,000 micrometers square, and I want a circularity between 0 and let's say 0.8. When I do that, you see some objects were filtered out and others were eliminated. Now, in many cases, you don't know this number exactly, right? But you can look at the objects and you say, yeah, these are the objects I want, right? So what I could do is I could reset this, and I'm looking for round objects, and I say, oh, this looks like a round object. So I'm not selecting objects one by one. I'm just updating the criteria here. So every time I select an object, my criteria gets updated. You see, when I clicked here, multiple objects got selected because I'm simply updating the criteria. So I've selected several objects that I thought were round enough, and I'm going to stop at that. Now I'm doing the same thing with the elongated class. I'm going to add area, and I'm going to add circularity. And here I want to select objects that are as elongated as possible. Maybe this one is probably not the best example for let me see if there are a few more. Yeah. So I've selected objects that I thought is elongated enough. Of course, if you have not selected the criteria very carefully, you might see some overlap between the two groups. But you see, these two groups are rather well separated out based on the criteria I specified. Now, if this criteria is not satisfying and it's not filtering it very clearly, you could keep adding more criteria. So you can make your criteria more and more stringent. You can even say, oh, I want an object not just elongated, but with a certain Ferret ratio. I want an object with a certain ellipse maximum, if I fit an ellipse inside there. I want an object which has a certain intensity as well. So you can add multiple criteria and make this filtering process really complex. Now, where will this kind of an application be useful? So maybe you are looking at cells in the metaphase or something like that, where you see you have normal cells in the metaphase. In the metaphase, the cells become if you're looking at nuclei will become condensed. The intensity will increase. So based on intensity and area, you can actually start filtering them. So a lot of applications for morphology and intensity-based filtering in image analysis. All right. I've done my filtering. The next step is a step I can eliminate. So the next step is actually doing measurements, right? So now I have to decide what kind of measurements am I doing. So I can always, area is one measurement I could do. I could measure the, let's say, the radius. I could measure intensity. I'm going to take the intensity mean value. And I could probably measure circularity. So that's a parameter we had used, right? So these are all the parameters that is going to be measured for the elongated class. Now I can, if I want, I can do some annotations. So this is just for visualization purposes. I can draw the center. I can draw a rectangle around it. I can give it a slightly different color. The same thing I can do for the other class. Area, radius, circularity, intensity. And again, some annotations. Maybe I give it a slightly different color. So these are the measurements I want to do for individual objects. Now, as I was saying, I can also do measurements on the group. So in this, let's say frame, I see, I don't know, 20 objects. So what's the total area of all the 20 objects? Right? What is the total area fraction that it occupies? So these are some of the questions I can ask for the group as a whole, right? So I can do some measurements for the group. Of course, there is a count. What is the total number of cells in the frame that satisfies that criteria? What is the area? What is the area percentage? What is the mean intensity of all these objects that you see in the frame? And so on. Similarly, for this group, I can measure the total area of all the detected objects, area percentage, mean intensity value. All right. Once this is done, I am now clicking finish, and this will finish creating the workflow. Now, you would have remembered that this is a time-lapse image, and this whole workflow was created using just one frame, and that was enough. Now what I'm going to do is I'm going to use this workflow to analyze the image. So every time I now have an image like this, I don't have to follow that rather long step. That's already saved. Only thing I have to do is I have to just click analyze. So I load the image, I load the workflow, I click analyze. So here it's, let me make it a little bit bigger. For every frame, you see it's detecting these objects. Detecting the round objects, is detecting the elongated objects, is doing all kinds of measurements on these objects. And it's also plotting a graph between radius and area. And you know, area is pi r squared. So if you plot a graph between radius and area, you should get a curve. But we have a small number of objects, so a tiny portion of a curve will look like a straight line. Let me stop this. Let's look at our measurements. See, all these measurements are done automatically. And I can have this group measurement. So in this frame, I have 18 cells. This is the total area of all the cells. This is the area percentage. This, let's say let me show one class at a time. The red objects or the round objects are occupying, let's say 3% of the total area of the frame. And this is the mean intensity of all these detected objects. And if I move this, you see this information, the group information for every frame is available here. And if I want individual information, this is information for every detected object in that frame. And this is what the measurement does. So this was a quick overview of image analysis or creating a workflow for image analysis. By the way, how much time do I have left? Dr. Bhattacharya, how much time do I have left?
I think it's like 40 minutes. You can continue till 12:30 IST.
12:30 IST. All right. Okay. Good. So I have some more time. All right. So this was about basic image analysis. Next, what I want to do is I want to show you machine learning based segmentation. So machine learning based segmentation is called Intellisense. So this is the Zeiss module or the Zeiss application for machine learning based segmentation. And as I was mentioning, the first thing in machine learning is training. Like my mother trained me to detect apple and orange. I have to be an instructor to the computer and I have to train the computer in detecting certain objects. And once I've trained the computer learns and then it starts segmenting itself. So here I'm going to create a new training model and let me call it HeLa cells. That's 23. This is my segmentation model. Here we are working a little bit differently. We are configuring the segmentation algorithm first before we do, let's say, pre-processing and other things. So then the segmentation will be done using machine learning. So I'm starting the training process. I say start training, and it gives me this nice graphic user interface. I'm going to select a file. All right, so this is my image. And you see, this is not a very nice, clean image like our, the fluorescent labeled nuclei. There are multiple problems here. First, there is a lot of unnecessary background. Then you see the images have not exactly been acquired at a good resolution. And, this region between the cells are a little bit bright. So a couple of issues with this image. So if I were to use a standard Otsu-based segmentation, this would probably fail. And that is why I want to try machine learning based segmentation. Now, the golden rule, or let's say the rule of the thumb in machine learning is computationally intensive because you have to extract multiple features and so on. So if you can segment an image with a more traditional approach, always go for the traditional approach because that will be computationally less intensive. You have to use machine learning only if your image is complex and you figure out that none of the standard methods for segmentation is not working. All right. So one of the first things I have to do here is to decide how many features do I want. As I was saying, in a traditional image segmentation, you are using one feature, you are using intensity, or you are using variance, or something like that, one feature. But in machine learning, you can use multiple features, like our example with apple and oranges. So when I look at an apple and orange, I'm not looking at just one parameter. I'm looking at, I don't know, 100, maybe a large number of parameters in my brain. So here I have the option of selecting how many parameters or how many features should you extract from the image to do the segmentation. The more features you use, of course, more accurate your segmentation should be, but then it becomes computationally more and more intense. And here you see we have two sets of features. We have basic features and deep features. Basic features are generated by filtering this image using multiple filtering algorithms. You could use a Gabor filter, you could use a Gaussian filter, you could use a Bayesian filter. Even for a given filter, you could use multiple parameters. And usually by using multiple filtering algorithms, you start extracting features. That is a traditional way of doing or extracting features. Now, another way of extracting features is called a convolutional neural network. So I will not go into the details of what a convolutional neural network is. I'm sure some of you have learned it. So this is also called deep learning. So we also have deep learning features, and there you can in fact generate as many features as you want. So you can extract up to 256 different features. So if you compare this to the standard segmentation where you are using just one feature, you can already appreciate this is a more complex process where you are making the system extract features and learn from those features. I'm going to keep it simple to start with. I'm going to start with basic features, 33 features. Now, as I was saying, you don't really need to understand the complexities involved. You just have to go ahead and paint on this image. Now, then I'm going to define how many classes do I have. I need, in machine learning, you need at least 2 classes. And of course, in image processing, you will have 2 classes. You will have at least one set of objects and the background. So 2 classes minimum. Here, I'm going to call my classes cells and the background. So I have cells, I have a background, and now I'm going to use a paintbrush. I don't know if you can see, I have a paintbrush which is orange in color. I can change the size of the paintbrush. When I change the size, you see the paintbrush becomes bigger. And using this paintbrush, I'm simply going to paint. I'm going to paint here, saying that this is part of my object. Paint here, saying this is part of the object. Paint here, saying this is part of the object. Now, you would have noticed that I'm not drawing the whole cell because this is a pixel-based segmentation or pixel-based classification. What I'm telling the software is these pixels belong to the object. And I'm telling the software these pixels belong to the background. And then I'm just giving it enough examples. I'm just, every time I draw, I'm kind of teaching the software that, see, please learn that, these pixels would belong to the background. These pixels belong to the object. Now, a rule of the thumb, or let's say a good practice, is not to train too much. Train a little bit because you don't want to overtrain. Then see how much the computer has learned, and based on where it has made mistakes, you can train specifically on those regions. It's probably also like training a human being, right? If you want to train, I want to train my son, I will train him a little bit, and I can observe where he's making mistakes and probably focus on those mistakes and train a little bit more. So I've trained a little bit, and then I can say train and segment. Obviously, it's going to make some mistakes. So yeah, it did mistakes, but as you can see, even with a minimal amount of training, it got a feel of what we are talking about, right? It knew that these regions are the background. Here you have objects. But one thing we did not tell it or train it is the boundary between the cells. So that's why we did not tell it, we did not train it. So it made mistakes there. I can go and refine those mistakes here. I can choose a smaller paintbrush. I'm specifically saying, you see this regions in between are background as well. Don't consider it as an object. Some errors here because these pixels were very similar to the background. So our algorithm made some mistakes. I'm correct. I'm helping it. I'm helping the software along as I train this more and more. And as you can see, this is a very simple process. You don't really need to be an expert in programming. Wherever there are errors. So I'm only helping with the errors. So one thing you have to observe is more I train, so this process called labeling. More labels I put, again, computation is going to start becoming more and more intense. So my goal is to use as little features as possible, as little labels as possible. So that will optimize my model, and that will help you complete the segmentation in smallest possible time, or let's say with minimal possible computational effort. Maybe I might want to train a little bit more in these regions here. All right, let's see what happens now. With machine learning, there's always a possibility that the system starts to learn in a very wrong way as well, because we have to test it out. Here, if you actually look at it, our situation improved from the previous situation, right? The space between the objects are now more clearly identified. It did some additional mistakes, which is fine. I can refine this. And each cell is almost nicely segmented out. Again, some more finesse is required. I can go ahead, train it a little bit more. Keep correcting the mistake. So that is the logic in machine learning training. Train a little bit, see where it's making a mistake, keep correcting the mistakes. Here's some errors with the background. Maybe a little bit more here. All right, let's see what happens now. Okay, it improved, right? As you can see, every iteration I do, the quality of segmentation is improving. Now we are very close to what we want, right? Small errors here and there. Keep correcting the mistakes. And with every step, the algorithm keeps learning. Of course, as I was saying, more labels I put in, it gets computationally more intense. Let's see what happens now. Okay, much better. Now you see there are still some errors, but these errors are fine because I have at my disposal post-processing, post-segmentation processing steps to eliminate some of these errors. And moreover, I'm not interested in all the cells. I'm going to pick some cells that I am very keen on. I'm not keen on all these cells that are on the edges and so on. I'm keen on these cells here, which are pretty nicely segmented, and the rest of them I'm going to eliminate in the post-processing step. All right. I mean, in fact, if you want, you can keep training a little bit more, but again, keep labeling as minimal as you can. And by the way, I had at my disposal more features if I wanted a more complex, analysis with the algorithm, but I've chosen to keep it simple. I'm going to say finish. So what I did now was to just create the trained model, right? I've not really done image analysis. To do image analysis, let me pull that image into my workspace. And as you can see, this was not really one image. This is a multi-scene image. There is a field of view with the stage was moved to different locations and different regions were imaged, and we trained it with just one image. Now what I'm going to do is I'm going to say I want to create an image analysis setting here. So the model that I have created will be used to do image analysis. So I'm creating an image analysis setting. Okay. And here you see the image analysis setting has been created automatically. My model name was called HeLaCells 23. The same name has been given to my image analysis setting. The two classes have already been automatically defined. I have cells and I have background. Obviously, I don't want to do much with my background. I will do most analysis with the cells. Again, the steps are similar. I say set up image analysis. I don't have to do anything here unless, So there is an option to combine Intellisense-based segmentation with, let's say, standard intensity-based segmentation. If that's the case, I could add multiple classes, but not required here. I would say next. I don't want to select a region of interest. And in this step, you see many of the blocks are missing. And they're missing because my segmentation is not done using Otsu. Segmentation is done using that model that I created. Of course, it has the post-processing steps like morphology, watersheds, but here I am preferring not to use them. In the next step, I'm going to create certain, yeah, I can change the opacity to make it look a little bit easier for you. I'm going to add the morphology filters. I'm going to say area and circularity like before. And now I can select some cells which are of my interest. And that's it. I say next. Here I can do my measurements. I can measure area. I can measure radius. I can measure what is called as the ferret ratio. Say next. By the way, if I want to put some annotations, that is possible. I can give it a different color. And finally, I say finish. Again, this workflow now is using that trained model, and workflow was set up using just one image. Now I've loaded that image. I have my workflow here that will call the Intellisense based segmentation into it automatically. Only thing I have to do is press analyze. As you can see, it takes a little bit longer than the standard segmentation or standard image analysis because we are using machine learning. All right. So I don't want to see the background. I only want to see and I want a graph to be plotted between radius and area. You can start seeing that curve. Area is pi r squared. You can see the measurements, and if I run this, you see the segmentation is done on all the images that were there in that. In every image where this condition is satisfied, you see the objects are detected. So as you can see with Intellisense, what we were trying to do was to make this process of machine learning more easy to use for users and avoid the need to do all this complex programming. All the programming is or complex algorithms are working in the back. And what you have was this easy to use interface, and coupled with that, this image analysis workflow, which is easy to set up. Now, every time you have an image like this, you don't have to repeat any of the steps. If you want to refine your training, so imagine you start acquiring more images and you say some objects are not being segmented properly, I can simply go and refine my training without having to redo my workflow. The same model will be used, same model will be saved with those changes, and your workflow will get adapted. So you can keep adapting your machine learning algorithm to new and new images that you acquire. Of course, if you are going to a completely different data set, from HeLa cells, maybe you are going to fibroblasts or something, maybe you have to create a new trained model. But otherwise, if you keep on acquiring similar kind of data, you can keep refining it. Okay, so this is the first and second part of what I wanted to show you: image analysis using standard method, image analysis using machine learning. The third part, I'm going to show you cloud image analysis based on cloud computing. And if there are any questions, I can take those questions now. Let me do a quick check of the time. Oh, I have 20 minutes.
Dr. Matthew, you can go up to 12:45 because it's going like an interactive session. So I think after your thing is completed, there will nothing left in the workshop because you are getting interacted with the participants every time.
Yeah, so, yeah. Actually, I might have to stop at 12:30 because I have another meeting after that.
Okay, sure. No issue. It's up to you.
Yeah. All right. Okay. So this was on, so all that we have done till now is on the platform, on the computer. Now I'm going to move to the cloud. How do I move to the cloud? I go to my web browser and I say www.appear.com. Now Appear is a free platform. I mean, in the sense that the basic version of Appear is free, unless you need complex computation, large data backup. Almost all the features on Appear are free. Anybody can just log on to www.appear.com and start using it. In fact, if any of you are on your laptops, you could even try it right away. But you have to go through a sign up process and create an account. And once the account is created, you get an interface like this one. All right, so this is my account, and you see there are 3 groups here. One is called the community content. The other one is called My Lab, and the third one is more information, which is if you want resources, if you want help, if you want to check the blogs and so on. Now, community content is, as I was saying, this is a community initiative. There are researchers around the world. There are people at Zeiss. There are people with some of our collaborators who keep creating modules and workflows, and they publish it. They publish it for anybody who is on Appear to use it for free. And that is community content. If you go to community content, there are a large number of modules. You can see the modules here. They're all created by different groups. So this first module here is created by Team Appear, which is our team in Germany. That is a Appear programming team. Here you see a module created by Abhishek. Abhishek is from IIT Mandi. He's working as an intern with us right now. There is one of other programmers at Zeiss who has created a module. Here you see a module created by a university. This is created by, in US, the National University of Singapore, Biophysical Fluorescence Lab. And there are different contributors. So as you can see, there are modules contributed by individuals, modules contributed by companies, engineers. Some of them who are part of Appear, some of them who do it because they like to do this in their free time. Some of the modules contributed by universities and so on. So a community effort in that sense. Now, every module, as you can see, is doing a specific function. So this is what is called as a label converter. This module is for segmentation. This module is for bleach correction. This module is for this technique called FCS, fluorescence correlation spectroscopy measurement. The module for background subtraction, module for changing the lookup table, and module for shading correction, and module for edge detection. So I don't know, maybe there are 100, more than 100 modules on Appear right now, and this number is growing every day as more and more people contribute. Now, if you log on to appear.com, all these modules, which are called public modules, are available, accessible to you right away. There is nothing more you need to do except create an account. Now, as I was saying, we can connect modules together to create a workflow. And some people have even done that. They have created these workflows and published the workflow. Let's find one of these workflows. So the workflow that I wanted to find is, this one. Let me go to the next page. Cell counter analyzer. So I showed you a video there, right? This is a public workflow. You see, it's using almost all the elements that we talked about in image analysis. The first module is a file conversion process because you might have a CZI file or something that is converted to OME-TIFF. OME-TIFF is the standard format that all companies now accept. OME stands for Open Microscopy Environment. This is like a society. This is a university-driven initiative. So almost any microscopy file format can be converted to OME-TIFF, and from OME-TIFF you can convert it back to some other format. So a Zeiss format can be converted to OME-TIFF, and from OME-TIFF you could maybe convert it to a Leica format or vice versa. first step. The second step is a pre-processing step. You are converting it to an 8-bit image. So if the image is not 8-bit, it could be 16-bit. So by bits, I mean, I was telling you that every pixel is denoted by a number. This number denotes intensity. So if you have 8 bits, you can represent 2 raised to 8, 256 intensity values. If you have 16 bit, you can represent 2 raised to 16 intensity values. But since we are doing segmentation, you don't need a very large intensity range. 8 bit is enough. So this is a bit converter, pre-processing step. Next one is another pre-processing step, auto contrast, which is increasing the contrast. This module is auto thresholding, which is a segmentation step. Fill holes is a post-segmentation processing step where after segmentation, there are sometimes some holes in the segmented objects. That's because maybe the intensity was not bright enough and those holes can be filled up. The third, the next module is a cell filter. So I showed you cell filters based on morphology. This is again considered a post-segmentation processing step. And finally, there is this cell report. So from images to report, this is an end-to-end workflow that you have create, use, and then share with somebody else if you want. So I can run this workflow, and there are two ways to run this. I can run it step by step, which means it will run one module, stop for my input, then run the next module and so on. Or the other option is to run in one go. That is, I call it as a single or a batch run. When I do that, it's asking me which file I should use. So that file is already on the cloud. So the cloud location is already provided. And I'm going to say run. Okay, the good thing about cloud is you can kind of do multiple things in parallel. Whereas if you have a software like Zen, you have to wait for one computation to complete before you start the next one, right? So I can create another window, www.play.com, and I can start creating a different workflow here. So here when I do, I can see that one of my workflows is already running. Here, I'm going to my personal space. This is my personal space. If you have your account, you will have your personal space. Here I have certain modules which are not public. These are some modules that I created myself, some modules that were shared with me by some of my colleagues or collaborators. I go to workflows. Here I have workflows that I've created, and I'm going to create a new workflow. And I'm going to call it yeast cell segmentation. So this is how I'm going to create this workflow. Obviously, you need an input image. So by default, the moment you say create workflow, the input block is always there. Oh, by the way, our this workflow is complete. You can view the results here. Here you see this is a segmented image. This is a graph that shows how the number of cells vary over time. And this was the output that was generated by the output module. I can close this, or I can say see results. You see all these results. If I go to my results section, the output of every intermediate module is saved here in my results section. So you have a very detailed documentation created automatically. And in many cases, you don't need all the intermediate results. If that's the case, I can just delete this with this delete button. If I want to take a look at one of the intermediate results, I can press this visualization button here. That was not. And if you want to share this file with someone, I can go and say share. It creates a shareable link. And let me try to post it on the chat here. So maybe you can try clicking on the link and seeing what I just analyzed right now. So it makes it very easy and convenient for people to share data with one another. So on the chat, I have posted this link. Please go ahead and try to click it and see if you can see this window. Okay, let it load. The meanwhile, let's continue with creating our workflow, right? So I have some E cells, images of E cells. So I'm going to do a E cell segmentation. So this is my simple workflow, only 2 blocks to start with. And I have to select a file, right? So this file I can drag and drop into my, so if I have, let me see if I have a file in my pictures. I don't have an image of E cells, but I have something else. I can drag and drop. And here you see that picture came here. Since this is not a picture of an E cell, I'm going to delete this, but you get the point, right? You can drag and drop images into this box here. Once you do that, all these images are saved on the cloud into your account. So I've already saved a lot of files in my account. So I'm going to select one of them. I have to go and find an image that says East cell. Maybe I can search for it. All right, so I have this image here. This is an image of E cells. And once I've selected the image, you see these 2 buttons went green, right? So if I had not selected an image, that is red. Red means some input is missing. So this could be, the input could be a file, this input could be a parameter. For this particular module, there is no parameter, right? So I'm going to say run. While this is running, I can open another window. A mistake there. And I can go and create yet another workflow. I'm going to call it rotate or image rotate. Same step, but this time I'm going to use a different module. This is geometric transformation. Here you see there are several parameters which I will have to fill up. I'll start with one image. So I'll go to, since this is a rotation process, I can select any image. So I selected a picture here. If you want to see what this picture is, I can see what that is. This is some segmented result of a material science sample. Yeah, so the image itself is not relevant, but what we want to do is to rotate this image by a certain angle. Yeah. Oh, by the way, my each cell segmentation is complete. These are my segmented E cells. Now, let me pause my rotation module for a moment. Let me go back to E-cell segmentation. And let me add another module, which is called E-cell analysis. So once you have segmented, you want to analyze it. And this is asking me for two inputs, the segmented image and the original image. The original image is coming from here. So I will take this wire and connect it there. So one input is coming from the first module. The second input is coming from the second module to this module here. Let me run this while this is running. Let me proceed with my rotation workflow. So here I've provided the image. Now it's asking me what kind of operation I want to do, right? I have not mentioned that. So I want to rotate. I want to rotate the image 45 degrees and I want the output to be ometive. If I want a different name, name this image Manoj if I want, and then I can run this. While this is running, maybe we can, okay, so my previous module is running as well and it just finished. Oh, I did not start that. So while this is running, maybe we can create another window. Now all this is happening on my account. So cloud has something called virtual machines. So which means instead of having one physical computer, it can create a large number of virtual computers and each computer can do this processing in parallel. That's why when I'm running these two modules, two workflows, both the workflows are running in parallel. Of course, this depends on how many workflows you are running, how many images you have. This is in the free version. There could be some limitations on how many things can run in parallel. But the biggest strength of cloud is if when you have a large data set, maybe thousands of images, and you do this simultaneously, the virtual machines can parallelize this process. That is significant gain in productivity or throughput in using the cloud compared to using the local machine. Now, as you can, you might remember the original image. You see, this is a rotated version of the image. This, by the way, is the result of my segmented E cell. So you see, this were the E cells. The segmented results are overlaid onto the E cell. There is also an histogram, which is a PDF. So PDF cannot be opened. I can download the PDF, and here you see there is a frequency distribution of the number of cells with a given area range. All right. So let me go back to my image rotate. And this time I am going to process multiple files in parallel, right? So I'm going to select this, this, So some may not even be an image, right? For example, this is CSV file, and I am going to process this. But because it's all happening in parallel, the one that is a CSV might just fail, but others will run successfully. There is an Excel file, which of course is not an image, that will also fail, but that is okay. I can keep adding images to this workflow. Just because one fails, that doesn't mean that the others will not work. They're all happening in parallel. You see, I've used different formats. I've used TIFF format, I've used CZI format, I've used OME format. There is a PNG format. So this module is designed to work with different image formats. I don't know, I might have selected like 20 different images. Now, when I say batch run, you see 24. So right now, let's see what the cloud is doing. The cloud is right now running 20 workflows in parallel. So these are all run through virtual machines. This 20 images, the rotation operation is happening in parallel for these multiple images. So this is definitely the power of the cloud. Now I can go here, I can view. So these are the 24 workflows running at this moment. 4 are pending because the server decided to keep 4 in pending and run only 20 in parallel. And that would change a little bit on how many users are there on the platform at a given point, what is the load, and so on. So as a free version, there will be small variations. So if you really want fixed or a constant amount of workflows to run in parallel every time, that might be a paid version that you might need to get. But otherwise, mostly for most applications, this free version is extremely good. Let me go and see what's happening. I can just close this. I can go to my results. So it's still working. In the meanwhile, let's look at a little bit of different ways in which you can load an image. So as I was saying, you can drag and drop, you can browse, and I can add an image here. So that gets added. Let me check my results again. Yeah, see the image rotate. One of them is complete. Number 5 is complete. Yeah, let it run for now. Okay, there are a few more things that we can see right now. So appear.com allows individual users to create modules. So if you are a programmer, if you're interested in programming, you can always go to the docs. Docs tells you how to create a program in Python, how to create a program using Jupyter Notebook. You can even use the GPU for computation. You can create machine learning based modules using TensorFlow. You can use Image or Fiji to create a module. You can use Java to create a module. You can use MATLAB to create a module. And all the examples are provided here. So if you're a programmer using one of these programming languages, please do go ahead, try creating a module, and then you could even choose to make it public if you think that module is useful for rest of the community. Now, many times what happens is there are people who are image processing specialists, but they don't get a platform to share the algorithms they have developed. So most of the work they do, it gets translated into an image or a figure in a publication, but they don't get so much recognition for the actual programming work they do. So this is a very nice platform. If you're a programmer, please do create modules, and that module will be useful. You will have, here I have, let's say, profile page. Here you can see all the modules that I created. Similarly, you will have a profile page. You can, let other people know the kind of modules that you are creating. Meanwhile, let's go back to results. Yeah, 2 of them are complete now. So usually maybe it's due to that the timing right now. The server is probably a little bit slow. Otherwise, this should have run slightly faster. And as I was saying, you can always share the data with someone. You have the option of sharing. There's also a new option called annotation. For machine learning, you have to say this is a cell and this is a background. That can be done now directly on Appear using this web interface, and then the labeled image can be downloaded, and this can be used for further machine learning based segmentation. So a lot of interesting options for doing this on the cloud. Let me take one final look at my results section. Yeah, it's taking a little bit longer than usual, but you see these images are being processed simultaneously. All right, I think I'm nearing my allocated time. right? So maybe I will stop at this point here, and if you have any questions, please do let me know. And even otherwise, if you want to contact me, you are always welcome to contact me for any questions, image processing, and to learn about machine learning, or you want help with Appear, you can always feel free to contact me. Yeah. So questions, if any.
Yeah, please come ahead if there is any other questions. So all participants are requested to come ahead and ask questions if there is any. I think they don't have. So any question like regarding confocal microscope, not only image processing I am telling about. For last 2 days, you have learned lots of things, I think. If there is any questions regarding confocal microscope, image processing, and other stuffs regarding imaging, please come ahead and ask.
Hello,
No?
this is Nirajan here. Hello.
Yeah.
Hello. Yeah. Does we can do this kind of experiments or do this kind of module work on the Z stacking image?
Yes. So each module is designed for a certain image dimension. So the modules that I showed you, they work on 2D data. Okay. But we have, so let me go back to up here. And if you go to public modules, you can search for 3D.
Yeah, okay.
So there is 3D volume localization, 3D skeletonize. There should be something called Stardust. So there are many modules that specifically work on 3D. But every time you use a module, there is, so if you are going to a module like this, there is some information here that tells you what kind of images it works with. So here it says it works with CZI and OMATIF, and it might tell you, it works with binary images, or it works with grayscale images, it works with 2D or 3D data. Here it says it works with both 2D and 3D data. So you can always read the description of the module. And in many cases, the person who created the module is also available. That name is there. You can contact that person and get more information. So yeah, again, to specifically answer your question, yes, there are modules with which you can do 3D segmentation and 3D analysis.
So is there any other questions?
Yeah, thank you.
Okay, so if any other participants have any kind of questions, please come ahead and ask. If no, then we can thank Dr. Manoj Matthew for his nice presentation. So hope the participants learned at least the basic of the image processing, how the image gets processed before the publication process in different reputed journals. So thank you, Manoj, for once again for your kind, informative talk and your time. So,
yes, thank you so much. I mean, it's great that you're organizing this online when people cannot meet in person. But I'm really happy to be part of this, and I'm hoping next time I could come in person.
We will be expecting that. We will be expecting that because our university previously, with the Association of Coral Eyes, conducted a workshop that is for only the basics of confocal microscopy imaging. Dr. Anupam Banerjee and other guys are also associated from your company's end. So definitely, hopefully in near future, when the normalcy came, we will surely plan to again for this kind of workshop, the advanced level workshop.
I will look forward to that. And dear participants, if you have any questions, you can always write to me. Please to go to appear.com. It is free, and again, I will always be available for any help or questions you might have. With that, thank you again, and I hope to see you all next time. Bye.
Okay, so with that, the technical as well as the hands-on session ends for this workshop. Now, there is an announcement regarding the certificate distribution, and before that, the feedback form. So we'd like to confirm you that the participants who have registered and who have filled the feedback form will be provided with the certificate, and the last date of filling the feedback form is Monday, that is 21st of September 2020. The participants having any kind of trouble while filling the feedback form, please contact us, and the email ID is provided to you in the flyer. So let me tell you once more, the workshop klyunib@gmail.com. So please contact us if you are having any kind of problem while filling the feedback form. And we would request you to proper filling of feedback form, because if there is any errors in names or email ID or the affiliation, then it will be remain as if you are filling over there. So we would request you all to take proper care while typing the Google form, that is the Google feedback form. And with that, we have to start the valedictory session. So first of all, we would like to invite Dr. Anupam Banerjee. I think he's present here from Carl Zeiss to say a few words regarding the workshop on behalf of your company. So Dr. Banerjee, are you present here?
Yeah, sir. Good afternoon, everybody. Good afternoon, everybody, the participant and eminent faculty members. So I'm audible, right?
Yeah, you are audible and visible.
Yeah. So it's a last 2 days, it was a very nice workshop. So in this scenario and in this situation, obviously, the online things are very important, and I think that workshop, all the participant as well as the speaker did it very, very well. And with the last 2 days, we came to know the different aspects of like the confocal research as well as the application part. And today, I think I also, though I'm from Zeiss, but still I learned many things from Dr. Manoj's talk. So those are very important. And the participants, those who are students, so to them, my request is don't forget all those stuffs within a day or two. Try to go to on more advanced level that we give you all those information. You try to go to your YouTube or maybe in the Google Chrome. You search our site. So there are many topics that you will get to learn about the confocal and how the application of the confocal will benefit you. So all those stuffs, and it is very, very good approach from the Kalyani University that on this COVID situation also they arrange such kind of workshops, so that will also spread a lot of knowledge onto the people who are working on the fluorescence as well as the confocal. And I'm very grateful to eminent VC sir, and obviously to the organizer, to Dr. Akash, Deepanjan and Dr. Vishal. So your all this effort was very good and very appreciating. And that's it from my side. Thank you.
Thank you, Dr. Banerjee, for your time deliberation now. So let's recapitulate what we did from the beginning. So if we start from the beginning, it was the inaugural session. So after inauguration, there was a keynote lecture which was delivered by Dr. Shah. Thereafter, Dr. Banerjee came and he talked about the basics of confocal microscopy, the instrumentation part and the principal part. Then there was a glimpse of the theoretical part, which was taken care of by Rishikant. He talked about the basics of digital image processing, and we called it for a day. And from today morning, Dr. Bhattacharya was there. He was talking about some specific applications of confocal microscope. He also showed how co-localization and all those stuffs can easily be detected using super resolution or confocal microscopy platform. Then Dr. Matthew came and he made us learn the ample amount of applications what a confocal microscope can do. So with that, I would like to thank the whole team, especially our Honorable Vice Chancellor for his constant support. Then I would like to thank Professor Rita Ghosh, Coordinator DST Parts 2. So without their consent, help, and support, it would not be possible for us to organize such kind of successful workshop. Then I would like to thank Carl Zeiss, our associated partner. So they helped us a lot. Their resource persons and other all those people who are very much helpful throughout the workshop. A special thanks to them also. Then I would like to thank DST Parts 2 for associated with us. And last but not the least, you people, the participants. So without your presence, this workshop is not a successful one. So with that, I'd like to conclude this for today. I think our Honorable Vice Chancellor and Madam is away from here because due to their busy schedule. So with that, I like conclude the workshop and I am closing it for now. So let's hope we'll meet in the future. And take home message is that this is just the beginning. Please practice and practice. It's like a practice. If you practice, you will be able to like grab good images as well as you will like to have good picture for process picture for your publication and so on. So as Dr. Banerjee told. So goodbye for now.
Thank you. Thank you all.
Thank you.
Hmm. Hmm.